computational model predicts phenotype from genotype, published in Cell, with Jonathan, the lead author, in 2012. And you were a graduate student at the time, right? Or a postdoc? Okay. All right. So you're up.
So I assume I need to stay here for the online participants.
No, they can see you if you walk around.
But the microphone is here.
Okay, no, there's microphones.
Oh, okay. All right, well, we get started. So thank you very much, David, for the kind introduction. I do like to think I was early, or myself and Marcus and our other colleagues were early. That said, certainly there are people doing whole cell modeling before me, and I think that's worth mentioning. In fact, there were other people modeling mycoplasmas even before me. In particular, Koichi Takahashi in Japan and his colleagues had developed models starting in the late 1990s of mycoplasmas. And they, in fact, collaborated with some of the same experimentalists which we collaborated with, who others in the room have subsequently collaborated with. And even before Koichi and Masaru Tomita in Japan, there were others that came much before them. But of course, things have evolved over time. Mike Schuler at Cornell in particular, as far back as the late 60s into the 70s, was already doing whole cell modeling. In that time, of course, it looked a bit different. This was before the rise of genomics. So the focus of modeling, the resolution was very different. But all that's to say that, in some ways, this is not a new topic. It's a topic that's been around for a long time, and the challenges have remained substantially for a long period. So I would encourage us to really think deeply about, given that some number of people have been thinking about these problems for decades, what new ideas, what new potential solutions can we bring to this that haven't already been explored over that somewhat long period of time? Although at no point was there ever really a large community, I think, over that time period. Okay, so what I'd like to introduce today is my thoughts on what I think are the most important challenges for the whole cell modeling, or for a emerging whole cell modeling community to address, and some early thoughts about progress that we've made at Mount Sinai towards some of those challenges. But first, I want to disclose, I recently, until a couple of months ago, I was assistant professor at Mount Sinai, where I had a research group which was pioneering whole cell models and methods for whole cell modeling. I recently left Mount Sinai to join Michael Antonov to develop Formic Labs, which is a company whose long-term mission is to develop holistic simulations of cells and tissues, and whose strategy in substantial part is to develop an operating system for life science research and development, with an emphasis particularly on modeling and simulation. I also want to thank all of the individuals who contributed to the ideas that I'll talk about this morning, particularly members of my lab at Mount Sinai and collaborators that we've had at Mount Sinai and beyond. Okay, so before we dive into how to build whole cell models and what the challenges are, I think it's useful for us as a community to try to come to some consensus on why are we even doing whole cell modeling, And what should the goals of wholesale modeling be? So I think we would likely quickly come to the consensus that our long-term objective is to develop comprehensive simulations that predict the behavior of individual cells or any arbitrary individual cell from microscopic information such as its genome. The challenge I see is that we're quite far from my vantage point from having that general capability of taking any arbitrary genome and predicting its behavior. And so I think it's useful for us to think about shorter term motivations, focus on a subset of possible motivations and applications for wholesale modeling. So from my vantage point, there are two principal applications which I think really drive the need for wholesale modeling. The first is precision medicine. We want to be able to deliver more precise, more personalized care to individual people. And that requires interpreting the genetic variation, which is unique to each individual person, and using that information to predict the trajectory of behavior or disease in individuals, and then utilize that information to design therapy which is optimized for individual patients. So from my vantage point, essentially what this requires is computational models that can account for genetic variation, which can occur in potentially any nucleotide in any gene throughout the entire genome, and combine that information together, encompassing multiple subcellular systems into holistic predictions of how disease could emerge in an individual. And then reverse engineer that information to design therapy. And so to my mind, this requires mechanistic models for us to be able to reason about how any arbitrary combination of variants collectively can impact behavior. That combinatorics, I think, is what, from my vantage point, what makes data-driven approaches to the same problem challenging. The second major application, which I think should be motivating wholesale modeling in the near term, is synthetic biology and metabolic engineering. But biology has unique chemical capabilities for synthesis and sensing, which I think we want to be able to harness to produce drugs, to produce commodity chemicals, to detect disease, to detect toxins. From my vantage point, one of the key bottlenecks is our inability to reliably design genomes to have particular phenotypes. And again, I think this requires comprehensive models that allow us to reverse engineer phenotype to identify genotypes that have particular desired phenotypes. Okay, so if we agree that these are two good motivations for wholesale modeling, the next thing I think we need to do is we need to think about what should a wholesale model be? What should it represent? What should it predict? How should it be structured? So I propose that we want to build models that capture entire cells, that capture entire genomes, that capture each gene down to the level of individual nucleotides, so we can reason about genetic variation, for example, or we can design concretely genomes to be synthesized for metabolic engineering. And I think we need to capture behavior over the entire life cycle of a cell, from birth to death and everything in between. Right? And as I've begun to introduce, I think we need to be thinking about mechanistic models, mainly so that we can use models to predict concrete interventions for medicine or concrete genetic designs for synthetic biology. And so to me, that means we need to be thinking about models which are capable of capturing the spatiotemporal dynamics of cells, and models which are capable of capturing the cell-to-cell variation among individual cells. Okay, so if we've established that this is our motivation and these are our goals, Now I think we need to start to think about how do we achieve wholesome models and what challenges do we have to navigate through. So in no particular order, one significant challenge that we need to navigate is what we're attempting to do is build comprehensive models which capture multiple subcellular systems inside a single model. This presents a challenge because these subcellular systems, which are existing simultaneously inside cells, these are operating at different scales, utilizing different physical and chemical mechanisms with different architectures. Historically, what modelers have often done is utilize different approaches that are tailored for different types of mechanisms or different architectures. And what we need to do is combine those historical approaches into, or one approach is to take those individual methods and combine them together. But that presents challenges from a mathematical perspective in terms of not having a rigorous mathematical foundation for such co-simulation of multiple mathematical representations. A second challenge is the sheer complexity of the models that we need to attempt to build. So a human cell has approximately 20,000 genes. However, by some estimates, there are at least 1 million distinct protein forms or proteoforms that exist inside a typical human cell. So the complexity of human cell biology is enormous. There's 1 million unique species, and those species interact together in a combinatorial number of ways, especially given the lack of insulation inside cells. So we need to be thinking about models which ultimately could potentially have upwards of 1 million variables, and which represent a combinatorial number of interactions, reactions, events evolving that 1 million plus unique species with unique chemical properties. Right. Another challenge is what we want to do is predict behavior by accounting for the microscopic mechanisms responsible for behavior. So we want to build simulations around the microscopic scale and then integrate those over many orders of length and time to produce predictions at the behavioral scale. This presents challenges for computational cost, which we then need to develop new computational methods to navigate. One potential way to address this is, and I introduced this from a biological perspective on a few slides ago, one potential way to address this is to take multiple mathematical methods, which essentially approximate multiple scales and combine them together. The challenge here is we don't have a mathematical foundation for how to do this rigorously yet. And more practically, we don't have great software tools to combine multiple mathematical representations together. More practical challenges include lack of any single experimental modality that we could use to provide data to calibrate or validate wholesome models. Instead, what we need to do is take multiple experimental modalities, which each provide snapshots of individual dimensions or sets of dimensions of cells and combine them together to train up whole cell models. Right? And worse, most of the experimental modalities that we might want to use to help train whole cell models, most of those experimental modalities produce incomplete data. So in a typical metabolomics experiment, for example, it's not possible to measure the entire metabolome of an individual cell with detailed temporal resolution. What's possible instead is to measure a subset of metabolites in a population of cells with very coarse temporal resolution without single cell resolution. So we have to try to compensate for this by using multiple experimental modalities, but that presents the same challenges that I spoke about on the previous slide. So how can we navigate these challenges? So myself and I think many others have tried to navigate these challenges by combining together multiple methodologies at each stage of model development and simulation to compensate for their individual weaknesses to build up a holistic paradigm for model construction and simulation. So in the data space, that means take multiple experimental modalities, multiple formats, combine them together. In data analysis, this would mean take multiple methods for reducing down different types of raw data, combine them together. In the model space, this means take multiple types of modeling representations, multiple simulation methods that are suitable for different subsystems and different scales, combine them together. So over the last couple of decades, I think myself and others, especially Marcus Covert and more recently Dan Schulten, who's participating in the meeting, have taken these ideas of integration and shown that it's possible to combine those ideas ad hoc and produce at least modestly predictive models of very small organisms. So this is an animation of the model that David referred to, that Marcus and I and others published in 2012. This is an animation of one in silico cell that was predicted by the model that we developed. The top left of this graph in blue illustrates the predicted dynamics of the growth of this one in silico cell. The top right illustrates the predicted size and shape of this cell as it grows, elongates, and ultimately divides. The middle left panel illustrates the predicted flux of each reaction throughout the cell cycle. The middle right panel illustrates the size of a protein complex that forms at the origin of replication to separate the strands of the chromosome to enable initiation of DNA replication. And the bottom two panels illustrated the locations of DNA binding proteins as a function of time and the expression of each protein monomer over the cell cycle. So what we've been able to show is that we can take this combination of methods and at least ad hoc, we can operate them at sufficient scale to build models that account for every characterized gene product inside a small cell. And we've been able to show that these models recapitulate a number of experimental observations. At the same time, there are a lot of gaps that remain in models, even of the very smallest organisms. So this is a graph which shows a comparison between model predictions in red and experimental measurements in blue for the growth rate of single gene disruption strains. So these are strains where one by one, we've taken each individual gene in the genome, we've deleted it, then we've simulated its growth both in silico and measured its growth experimentally. And then we've plotted both the predicted and measured growth rates in red and blue. And what you see is for most disruption strains, the model is pretty accurately able to capture the growth of this organism. At the same time, there are several cases here and here and here, for example. So, but there are several cases where the model is not accurately able to capture the contribution of genes to behavior. And this is for the very smallest organisms. So what we take away from this is that there's still substantial work that remains to build accurate models, even for the smallest organisms. And there's a lot of work that remains to do to scale up this methodology so that it could be applied to much more complex organisms. And so that's really the central problem that my research group had been focusing on Mount Sinai over the last several years is how do we build methodology for building much more accurate models and models of much more complex organisms, ultimately including human cells. So what I'd like to do in the remainder of the presentation is quickly walk you through some of the methods and tools that we've developed, and then I think we'll reveal a lot about how we've been thinking about this problem of building wholesome models and simulating them. So one of the key problems in wholesome modeling is we need to gather data that we can use to train very high dimensional, very large models of cells. So let's just think about metabolism for a moment. Let's say that we wanted to model the dynamics of the metabolism of an organism. At a minimum, we would probably want information about concentrations of metabolites, information about abundances of enzymes, information about the kinetics of reaction or kinetics of catalysis of reactions by enzymes. And where this gets challenging is there's no single experimental modality that we can use to measure all of this, let alone measure it simultaneously. There's no single database, no single paper that you can just go to obtain all this information, especially not comprehensively for any organism. Instead, what you could at the time attempt to do is utilize multiple resources, work with multiple websites, multiple APIs, multiple formats, multiple different formats for data, multiple different ways of describing metadata. You could work with all this complexity or chaos and try to navigate through all of this yourself. You could, when there's missing data, try to find data for similar molecules, similar reactions in maybe closely related organisms, but you have to navigate all of this yourself without any direct support from any of the tools or papers, search engines that you might use. So we felt a better approach is let's try to centralize the most common types of data that would likely be used for wholesome modeling, and let's build better tools that enable us to better leverage the data that exists to help us find relevant data to any modeling project. And if we do things this way, the idea would be rather than trying to find data for every modeling project, so every research group that's trying to find data for modeling, instead of every research group and every graduate student try to solve this problem themselves, substantially repeating the same work, we would try to centralize this, do it once, do it really well, and then just make this available for everyone. And ideally kind of grow this into a community project where the community was contributing to this great data resource. So we developed Datinator. It's a toolkit for finding relevant data for biochemical modeling. It's available at datinator.info. At the core of Datinator, we've integrated multiple types of data which are relevant to biochemical modeling, such as concentrations of metabolites, abundances of proteins, half-lives of RNA, half-lives of proteins, and so on. And then on top of this, we've built a search engine to make it easier to find relevant data for any modeling project. So if you come to the homepage, you're asked two questions. You're asked, what biochemical mechanisms would you like data about? And what organism would you like, or what genetic context would you like that information? And then, like other search engines, we start by trying to essentially disambiguate your query, and we search our database for metabolites, genes, and reactions which potentially match the biochemical mechanism that the user queried for. And if a user then drills down on any one of those metabolites, genes, or reactions, and they're presented with tables of different types of information that's relevant to, or potentially relevant to, that molecular mechanism in the organism that the user queried for. But rather than only pulling down information that is specifically measured for that particular metabolite or gene or reaction in that one organism that the user queried for, instead what we do is we build clouds of information centered around that molecular mechanism and organism. And the reason that we do this is because for any individual combination of mechanism and organism, we often don't have any relevant measurements. And so what modelers are typically doing is using other relevant data, but they're doing that ad hoc in ways that leave a lot of data on the table. So we're trying to do this systematically, build such clouds of data. So if we search for a particular metabolite, we're going to pull down data not only for that metabolite, but also data for structurally similar metabolites. And if you search for a particular organism, we'll pull down data from all other similar organisms. And then what we do is we provide you tools to be able to filter down or constrain those clouds so that you can narrow down on what you feel to be the most relevant data to the system that you're studying, at which point you can incorporate other background information, which is perhaps difficult to systematically encode. So once you've narrowed down that cloud, you can then export that for incorporation into modeling pipelines. So over the last several years, we've developed a bunch of additional tools for wholesome modeling. We've developed tools for systematically incorporating data into Datonator. We've developed, I'm going to go through this really quickly because they're running short on time. We've developed tools for concretely annotating the semantic meaning both of data sets and models. One of the key gaps in our ability to annotate meaning was ability to describe physiological macromolecules that participate in biochemistry. These are typically identified with identifiers like for UniProt or FASTA sequences, but this emphasizes the chemistry of unmodified macromolecules. And to be more precise, we need to be able to describe molecules that have modifications. So essentially what we've done is generalize what is typically known as FASTA to be able to account for the chemistry of physiological macromolecules. And then this can be used to either annotate data sets or annotate models, and then further used to identify gaps in models, missing species, opportunities to combine models. Another problem that we've needed to solve is we've needed to make it possible to systematically describe composite hybrid models. To address this issue, we developed a new language called WCLang and associated simulation tools. The last thing that I'll describe is we felt that collaboration will be essential to development of wholesome models. So we've developed a few resources to facilitate sharing and reuse of models and simulation tools. Biosimulations is a central repository that we've developed for publishing models. We started biosimulations by integrating simulation projects, which includes models, simulations, simulation results, and data visualizations of simulation results from several existing model repositories. And now users, the community, can also publish models here in a wide range of formats. To make it possible to have a centralized database that supports multiple types of models involving multiple simulation methods and multiple simulation tools, we developed a web application called Run Biosimulations. To users, what you get is a web form which you can use to simulate models in a wide range of formats with a very simple point and click interface. And to make that possible, we needed to build a registry of simulation tools, which contain tools with standardized interfaces, so that once you learn one set of conventions, then users have the ability to execute any type of simulation and use APIs consistently to integrate them into more comprehensive simulations. As part of biosimulators, we also have a web-based interface for navigating this registry and finding simulation tools that are appropriate for particular simulation studies. So in summary, we believe that one of the potentially the best way to approach whole cell modeling is to take many individual existing approaches and combine them together to complement each other's weaknesses to enable comprehensive simulation. So in the space of data, we think that means combining together multiple experimental modalities. In the space of modeling, we think that means combining together multiple mathematical representations. In simulation, that means combining together multiple simulation algorithms and mixing all this methodology together. And going forward, I think where the whole cell modeling community needs to focus a lot of its attention is not only doing this ad hoc, but working out ways to do all of this systematically so that it can be applied at sufficient scale to produce holistic simulations, including of complex organisms. With that, thank you very much for your time and attention.
Quick virtual round of applause as they get set up. What we'll do is we'll ask a few questions and I think probably take some from the floor in Chicago. I see Elizabeth has just put in a question in the chat, and actually Robert and Wallace and Aladdin have come in with questions as well on Keystorm. So thank you very much indeed. What I'll do is I'll wait for Jocelyn and Effie to get sorted, and then
we'll do a couple questions in the auditorium, Toby, and then on and to you for the virtual questions, right?
I think that sounds splendid.
Okay, so if you have a question, we'll give you a mic. Just raise your hand. I will start very quickly to a trivial question. Jonathan, Datanator, that's available publicly?
Everything that I described is available.
Yeah, maybe you can post a link for us to go to. And the second question I had was, you certainly gave us a lot to think about the complexity, the million members of the proteome. But beyond that, there's also the milieu to make it worse. But I mean, proteins can behave quite differently if they're embedded in a membrane or in the cytoplasm, and many move back and forth. And I'm just wondering if that's something you're also thinking about or not now.
I mean, certainly it's on our list of challenges to address. I think others have focused more attention on that particular problem, however.
Any questions? Right.
Hi, Jonathan. In your cartoon, in one of your later cartoons, it looks like things are mostly about the cell cycle, DNA, RNA, protein, and everything. So I have two questions. One is, do you try to model, when you're doing whole cell modeling, do you try to model every single interaction, like every protein-protein interaction? And then my other question is, what do you do, for example, about proteins whose functions are not well understood?
Yeah, great questions. So in principle, we would like to be able to capture every protein-protein interaction. Realistically, of course, we're not presently doing that today. Presently, we're focusing on protein interactions which have known chemical consequences or known functions. And in terms of proteins which don't have clearly annotated functions, what we do, I think really the main thing that we can do is we can account for the metabolic cost of synthesizing their RNA or synthesizing the DNA that encodes them, the RNA that expresses them, the protein that expresses them. We can, to the extent that is known, we can capture their composition in protein complexes, but their function, I think is difficult for us to account for. And that I think represents one of our opportunities and challenges to improve models. I think if one wants to try to simulate the functions of unknown proteins, then one needs to focus on lower scale simulations, which is possible. I've been conceiving that as the domain of maybe structural biology rather than the domain of whole cell modeling. Or at least that's where I've positioned my own research group, just because I think you can only go after so many. But any one research group only has so much bandwidth.
Toby, can you hear us?
Yeah, very faintly.
All right, so we're ready for the virtual questions.
Well, I think we've got actually a host of them. We may not be able to get through all of them, but I was wondering if I can first pick off Robert to kick off, and then go to Wallace, and then go to Elizabeth. Robert first. Do you just want to read out your question, or I can do it for you?
Sure, I can do it. So my question, thanks for the really nice talk. So my question is, in the model the data you showed, you compared the model of different mutants. It looked like to me that the model recapitulated quite well the different mutants, but one thing that caught my eye was that the variation, their variance, that is the size of the error bars, was not well recapitulated by the whole cell model. And maybe you could just comment on that and give us some ideas as to why you think that might be. That is why it's not capturing the variation in different mutants. Thank you.
Well, I think one of the perhaps the bigger issues there is the experiments are not very precise. So I wouldn't look at that as that's not reflecting the variance in the growth rate of individual cells. That's reflecting our inability to precisely measure the growth rate of those strands.
Yeah. Mm-hmm. Thank you.
So it's not something that we're trying to capture. We're not trying to, our goal wasn't to try to model the imprecision in our experimentation.
So it's not built into your model of variation of growth within a population, or is it, or is it come out of the?
So I think, so what's of interest to us is to try to capture the variation in the growth of individual cells, but those error bars are not reflecting that variation in individual cells. They're reflecting our imprecision of measurements. So these are two different things. And in mycoplasmas, because the cells are very small, it's hard to do single cell experiments. So at least at the time, we just didn't have information about the variation in the growth of individual cells. That would be nice to have. I think that's, I think the easier way to address that problem is to model other organisms where it's easier to do single cell experiments rather than to try to develop methodology for single cell experiments for mycoplasmas.
Robert, with your permission, I would like to get at least one other in from the virtual team. Wallace, you had a really interesting question. Do you want to share that?
Yes, so my question is, I guess, about what is the right scale of abstraction. So, and I was thinking about computers as an analogy. That's a case where, in contrast to the cell, we understand every part of the computer exactly. There's no imprecision or confusion, and yet nobody would ever try to do a whole computer simulation. It just isn't done. And instead, computer science has worked because people have picked much higher levels of abstraction. Register level description, gate level description, register level description, finite state machines, and so on. So do you think, how convinced are you that sort of the protein level is really the right level of abstraction for modeling the function of a cell? Because like transistors would be the wrong level for computers for sure.
I think one of the big differences is that in electrical circuits, there's insulation. And that insulation is what I think enables abstraction. And
I don't agree with that, actually. I think the insulation allows you to modularize and model individual parts. The abstraction sits on top of that. So even if you made digital logic in a way that was not modular, it wouldn't actually make any difference when you go to the next higher level.
But I think from my perspective, the difference is that biology is not modular in the same way.
But I don't think it needs to be modular. I think if electronics, so okay, analog circuits are also not modular, right? But you can, again, you model them, no one tries to model, a complicated analog machine transistor at a time. You basically break it, you say, well, what I'm trying to make is an amplifier, and then you model, the frequency response of your amplifier, and that's where you do your design. So I'm not sure the modularity is really irrelevant. I mean, I get your point, and I totally agree that is a huge difference. But I don't know. I think that tells you how you handle any one level, but I don't necessarily think it means you can't abstract to the next level up. I'm not saying you're wrong. I'm just saying I don't know. Maybe you're right.
From a practical perspective, today, I don't know that starting from the level of that abstracting molecules really is sufficient. That's where I focused my work, because I think there are so many challenges that one can't attempt to build atomistic simulations as a small research group. I think it's just not possible to try to work from that level all the way up to behavior and do so on physiologically relevant timescales. I think ideally
You're already using a level of abstraction, essentially.
Well, yeah, I think you're forced into that as a practical matter in order to try to make progress. I'm not doing that because I believe that that's necessarily the right way to solve the problem. If I had a much bigger budget, or we could all agree to work more cohesively, I think we could maybe work out a way to do this.
Got it. Thanks.
Okay. David has a solution to the problem.
One more virtual? Okay, Toby, we can take one more virtual and then we'll move.
Well, it's a nice quick one. It's from Elizabeth Haswell, and she can't speak because she's got a very noisy background, and it's a nice simple one, which was, Jonathan, that you had 4 organisms represented on the detonator. What were they?
There's a question about what was in one of the slides.
Well, I think it was 4 organisms. That this is Elizabeth's question. There were 4 organisms represented, and I think she was curious as to what they were.
I don't remember which slide this is referring to, but Datainter has data for many organisms, much more than just 4.
Very good.
The data is heavily skewed to some organisms. I'm not sure if that's so perhaps not unsurprisingly, the content of Datainator is heavily skewed to humans, mouse, yeast, and E. coli.
Well, perhaps if there's another one, which were nice and simple one, Aleph Beisov, you came up with a nice simple question. Do you want to just repeat that for us? Aleph, you're muted at the moment. I might just read it for Aleph, if that's okay, Jonathan.
Toby, she's actually in the audience, so she's here. She's gonna say it out loud.
Oh, lovely. How smashing. Thanks a lot.
Hello. So my question was that you mentioned that we can't measure all of the metabolites in a cell, and rather that we focus on specific subsets of those metabolites. So my question was, how do you determine which subsets are most informative? And could integrating those subsets kind of give you somewhat accurate of a prediction of the metabolites function within a cell?
Well, so I was referring to metabolomics experiments, and in those experiments, it's not a targeted investigation of a particular subset of metabolites which are chosen ahead of time. There's only a subset of metabolites could be disambiguated. So the methodology doesn't require the investigator to ahead of time decide which metabolites one is going to attempt to investigate. you could go that way and develop biochemical assays for detecting individual metabolites. That's of course a lot of work to go that route rather than the more unbiased route.
Okay. Jonathan has lots of questions to answer here. So yes, I'll ask you a quick one. Well, no, it's not gonna be, I'm sorry. So it's actually a follow-up to something that Wallace said, which I thought was a really interesting one. So I saw on your biosimulators thing that you support things like BioNetGen. And when you make a BioNetGen model or a model in rule-based languages like that, combinatorial complexity can really bite you, right? So you can get models that can theoretically generate like 10 to the 23rd separate species. And the only way we know how to simulate those things are using these relatively slow agent-based type simulations where rather than sort of enumerating those species, you sample them. I'm just curious if you've thought at all that that sounds like it might slow things down if you tried to use it, right? Like, or maybe if you try to use a BioNetGen thing and you went in naively, you might be like, oh my goodness, I can't use ODEs for this, so I'm sort of screwed. Have you thought about it? Is there any way that you can think of, or if you thought at all about how to sort of automatically course paint some of that stuff? Like focusing on just the things that matter for the other stuff you're trying to hook up with. Maybe there's no way to do that, but I was just thinking maybe you'd thought about that because sometimes those simulations are so slow, you couldn't ever do what you want to do.
I focus most of my attention on a different dimension, which is let's just try to incorporate as much mechanism as possible because I feel like we don't know what's important, or there's always counterexamples which would show that, like, the reason that the mechanism was discovered is because there's typically some scenario in which it is important. So what we've philosophically decided is that means we should try to incorporate it into a model. I think part of your question also is there are reasons that I like rule-based modeling. I think it's a nice solution to capturing the combinatorial complexity. It does introduce computational challenges of computational cost, but what I think discrete simulation does is it makes model composition easier. So you have this trade-off of like, it's easier to do one thing, but it makes computational cost higher. On the balance, because of the ability to facilitate model composition, I think it's a promising strategy. I think that the continuous simulation is much harder to do model composition. And I think discrete simulation also lends itself more easily to parallel simulation approaches. So you don't lower the computational cost, but you can lower the wall clock time at least at more dollar cost, actually. So there's trade-offs. I think actually this gentleman in the blue shirt has been raising his hand patiently. What was the rate that you're referring to? We don't have any corresponding experimental data for that. And that part of the animation is, to be honest, is it's not something that's really tied directly to simulation. It's just something that we added as only part. For the most part, without exception, it's all based on experimental data, but the shape itself is something that's just part of the animation. That's where we're cheating a little bit.
All right, I think we should,
The rest is real.
credit. Thank you. We'll ask Jonathan to answer the rest of the questions on KaiStorm. I know there is more hands that were up and really sorry we can't take all the questions, but please go to KaiStorm posted and we'll have Jonathan maybe go and answer those.
Actually, we're going to takes a minute to transition on this complex hybrid meaning. So while that's happening, I'm going to get in the last question. What about AlphaFold? They just dropped 200 million structures. Are you guys, Jonathan, are you guys thinking about using that? That seems like a really great source of data. Yeah.
We have not. We've been discussing using.
The answer to that was no.
Although at Formic Labs, we do talk about structural biology and AlphaFold fairly frequently.
The answer was at Formic Labs, they do talk about structural biology. Okay, so I am going to now introduce a new feature of the FYM. If you've come to these meetings before, we haven't had a small breakout discussion groups. We're doing that this time for the first time. We're incorporating a theme. The idea is this theme will run throughout the meeting. And you know what the theme is. The theme is modeling whole cells. I want to just give you a little background. How did this come to be? How did we end up with this? So it started back in November last year. Elizabeth and I met with our new KI team, Jocelyn, Effie, and Toby. And I met Toby for the first time. You've met him now too. And you realize that he's a just adorable, charming Brit. And we were talking to him about our meeting and he was taking it all in and he was saying, yeah. And just imagine him saying this in his charming, adorable Brit accent. David, you have many nice features. I can't do the accent, but you have many nice features. But could we just take a moment to have a cup of tea and take a moment to think? Could we do a theme? And that's like, what do you mean a theme? The theme is we have a community, we're developing, we're trying to get collab. That's the thing. And he said, no, I mean a theme to go throughout the meeting. And so we talked about that and I sort of got what he wanted. And then Elizabeth came up with the theme. Modeling whole cells. And I said, yeah, that's a great idea. I mean, it's something I thought about a long time ago and never thought would actually happen. So meanwhile, Toby is stroking his chin and we talk about this some more. We decide that this is a good idea. At least let's run it by the steering committee. Now, remember I said this is an RCA grant from NSF. In their wisdom, they require a steering committee. And so we utilized them. In January, we held a Zoom meeting with the steering committee. We pitched this idea of a theme in this meeting, whole cell modeling. So on the steering committee, we have cell biologists, Liz, John, Suzanne, Julian, Ment, and Rob. And their reaction, as I remember, it was more or less, yeah, whole cell modeling, far out, man. They liked it, more or less. On the other hand, our computational experts, Belinda, Wallace, Carlos, and Wouter Jean, they didn't not like it, but they were much more cautious. Like they already started resonating the challenges and the difficulties which Jonathan has just laid out for us. But at the end of the meeting, they did cough up some names, one of which was Jonathan Karr, and the other was Zahn Luthi Schultz at UC, University of Illinois, Urbana-Champaign, who we'll hear from in our second keynote tomorrow. So that set me off on my search to learn about Jonathan's work. That's where I found that paper, the cell paper, and with the 1,404 citations. And I requested a Zoom chat with Jonathan, which he very generously agreed to give me. And we chatted one afternoon. And I was thinking, I'm going to tell this guy who's the father or at least co-parent of this field, even though then I didn't know that there was all this history before his paper. I was looking at him as they had sort of started it. We're going to do a meeting on your field, on your child. I thought he would be like very excited. Well, he was interested and happy to talk to me, but what he spent most of the Zoom talk informing me about was not just the difficulties that he outlined here, the challenges of doing this, but it was the practicalities in the real world. He talked about funding. And one of the things he said is you cannot put a grant into NIH or NSF to do whole cell modeling. And the reason is it will go to a regular panel and it will compete with other grants that are not doing whole cell modeling. It's not a fair playing field. He seemed a little bitter, I have to say. And so he gave me this reality to think about. And so came back to the meetings with Elizabeth and KI, and we decided we're going to go ahead with this. But what we want to do is free you of this constraint of the real world. So as of today, I am announcing that Congress has allocated $10 billion. There's your funding over 10 years to create something the government is calling the National Institute of Whole Cell Modeling, NIWCM. And President Biden has appointed you to be in the 8-person board of directors. And over the next 2 days, you're going to meet with your board members 3 times, and you'll begin discussing how NIWCM will accomplish its 10-year mission. And I think that Jonathan has really redoubled my appreciation for the challenges here, but I just want to remind everybody of the obvious. This might be something of a moonshot, but in 1960, President John F. Kennedy said, before the decade is out, we will put a man on the moon, and we did. And at that time, I don't think NASA could get a rocket not to explode on the launch pad. I mean, we were not even a suborbital at that time. And in less than 10 years, we landed a man on the moon. So I think with the $10 billion I'm giving you, you should be able to accomplish it. I'm expecting it. All right. So with that, I'm going to turn it over. Yeah.